Hello friends, today's topic is autoclave. In this topic we will understand the working principle of autoclave and types of autoclave. We also take a look on biological indicators and its use in calibration. History of autoclave. The autoclave was reinvented for medical and scientific use by Charles Chamberlain in 1879. Do you know how an autoclave works? As we know that autoclave is used for the sterilization of inanimate articles. The working principle of an autoclave is simple. We use water in autoclave, and with the help of heating coil water is boiled and pressurized steam is generated, which act as sterilization agent. As temperature of water increases, the saturated steam produced under pressure. Moist heat destroys microorganisms by the irreversible coagulation and denaturation of enzymes and structural proteins. For successful sterilization cycle, 4 parameters of steam sterilization are important. First parameter is steam. The moist heat is lethal than dry heat. It penetrates in the cell and destroys microorganisms by the irreversible coagulation. Second parameter is pressure. A very high pressure, that is 15 psi, rupture the cell. Third parameter is temperature. High temperature kills microorganisms rapidly. Fourth parameter is the time. Time plays important role in sterilization process. Exact time is important to achieve perfect sterilization. Less time cannot sterilize material, while excess time may denature material, so time is most important parameter. The 2 basic types of steam sterilizers are: first, gravity displacement autoclave, and second is high-speed per vacuum sterilizer. Remember that steam is admitted at the top or the sides of the sterilizing chamber, and because the steam is lighter than air, this air forced out from the bottom of the chamber through the drain vent. Let's see types of autoclave one by one. First type is gravity displacement autoclave. Type of autoclave used to process laboratory media, water, pharmaceutical products, regulated medical waste, and non-porous articles whose surfaces have direct steam contact. In gravity displacement autoclave, when the steam enters in nth sterilizer chamber, it displaces the residual air through an open vent. Specifically the per vacuum cycle, in which the air is removed from the chamber and loaded by means of pressure and vacuum excursions. For gravity displacement sterilizers, the penetration time into porous items is prolonged because of incomplete air elimination from load. High-speed vacuum sterilization. The high-speed per vacuum sterilizers are similar to the gravity displacement sterilizers, except they are fitted with a vacuum pump or ejector. To ensure air removal from the sterilizing chamber and load before the steam is admitted, the air is mechanically removed from the chamber and load through a series of vacuum and pressure pulses. This allows the steam to penetrate porous areas of the load that couldn't otherwise be reached with simple gravity displacement. Now we know that how autoclave work. So let's know the operating procedure of autoclave. Open the lid of the autoclave by pressing the steel base at the bottom. Remove both the perforated steel baskets from the autoclave. Pour enough purified water into the autoclave such that the heating coils are completely submerged in water. Replace the stainless steel baskets back into the autoclave. Load the material to be sterilized into the basket. Close the lid and clamp the screws in place. Autoclave process monitoring. Unlike the other, autoclave process is also monitored. In autoclave, the steam cycle is monitored by mechanical, chemical, and biological monitors. In mechanical monitoring, the printouts of cycle are taken and verified the time, temperature, and pressure parameters. Monitoring is also done by using chemical indicators. These chemical indicator change its color when exposed to specific temperature and pressure. Autoclave's efficacy is monitored with the help of biological indicator. In the next slide you can see the photos of forms of the biological indicator. Let's understand the biological indicators. We will use this biological indicator in calibration of autoclave. In simple language, biological indicators is a test systems which containing viable microorganisms and we know its resistance to a specific sterilization process. Generally biological indicator are nothing but the spores of resistant bacteria. A known population of spores of resistant bacteria are sticked on paper strip. The strips are carefully packed. In case of vials, the known population of spores of resistant bacteria with dye indicator and growth media is packed in the form of vials. Use of biological indicators depend upon sterilization cycle and sterilization method. For steam sterilization, Geobacillus stearothermophilus is used as biological indicator. For dry heat sterilization, Bacillus atrophius is used as biological indicator. Biological indicators are come in variety of forms, such as spore strips, ampoules. Each one has its own working principle. Why we use biological indicators? Biological indicators provides information on whether necessary conditions were met to kill a specified number of microorganisms for a given sterilization process. It prove a level of confidence in the process. If we sterilize the biological indicators and incubate them for specified time, then there should be no growth. If growth present, it means the autoclave cycle is failed to sterilize material. Generally biological indicators used in calibration test only. In routine process for monitoring the cycle, chemical indicators are used. First, take 4 strips of Geobacillus stearothermophilus spores of population 1 million. 2. Take 5 tubes containing 100 ml sterile soybean casein digest medium. Henceforth it is called as SCDM. 3. Label tubes: 1 tube as negative control, 1 tube as positive control and 3 tubes as test 1. Test 2 and test 3, 1 tube as labeled as positive control tube. 4. Take 3 biological indicator strips. Place 1 strip at drain point of autoclave, 2nd strip at middle of autoclave and 3rd strip at opening of autoclave. 5. Start the autoclave cycle. 6. After completion of cycle, carefully remove the strip and take it to laminar airflow. 7. Open the strip cover and aseptically inoculate strip in 3 tube. Labeled as test 1, test 2 and test 3. 8. Negative control tube should be keep as it is. 9. Keep 1 strip unexposed for positive control. 10. Incubate all tubes at 55 degrees Celsius for 7 days. Thanks for watching this video.